Namaskar dosto, welcome to channel Toxicology on board. Today in this video we'll be discussing some of the important topics. These are not from ABT recertification question papers, but these are given from the senior most toxicologist in the fraternity, like Dr. K.S. Rao or Dr. P.K. Gupta. So we'll discuss question number 6. Question number 6 is which of the following can be used to characterize a response to a toxicant. So something which is related to I think second or third chapter in the case of a Clayson textbook, and under ABT brochure 2026, page number 25, as well as in page number 26, they have given this topic like application of omics technology appropriate to test a hypothesis under investigation. So a new type of toxicological sciences, omics will comes into omics will and its application will be there in case of prediction as well as in computational toxicology. So one question with respect to this is given. So which of the following can be used to characterize a response to a toxicant? The right answer will be in the green text, that is metabolomics. Proteomics is for proteins and transcriptomics is for RNA. And so you can pause the video and look into this picture. I'll show you another in the next slide also, taken from a Clayson textbook. So first let us understand. When I was going through this question, I was a bit confused between what is the difference between metabolomics as well as metabolomics. Okay, so we'll very quickly go through what are the differences there. Okay, at the time of you reading either place in third chapter, I guess second or third chapter it is given. So we'll see while metabolomics and metabolomics are often used interchangeably, like Noel, how we'll be using like that. They reflect like PDAD. They can be interchangeably used. They reflect slightly different scopes of study. So the scope of metabolomics is little bit different when compared to metabolomics. So metabolomics focuses on comprehensive profiling of all metabolites. Here you can see metabolites within a single cell, within a single cell, a tissue or organism. Whereas metabolomics specifically measures how a complex multicellular organism metabolism changes over time in response to disease, drugs, or environmental stress. This is taking metabolism is taking at a single point of time, but this metabolomics is taking over a period of time. Okay, so here you can see the scope. Metabolomics focused on cells, organs, as well as isolated system, whereas in the case of metabolomics, it is system biology approach focusing on whole complex multicellular organism. Primary focus of this metabolomics is identification and quantification of all metabolites, whereas in the case of metabolomics, dynamic, time related. As I told you, know metabolic responses of an organism to a particular stimuli. Okay, so generally metabolomics you are studying by using mass spectroscopy, whereas metabolomics you are using nuclear magnetic resonance spectroscopy. Okay, so I understand not many of us, this will be mostly done by computational toxicologist only. Not every toxicologist will get an exposure to work on this type of all proteomics, metabolomics, transcriptomics. So this is the diagram which is taken from Clayson's 9th edition, page number 49. So here they have given the concepts of this omic. So conceptual approach for incorporating omic technology and resulting large databases into toxicological evaluation. See, whole point of this type of omics technology is computational technology, bringing the information from proteins, from the DNA, from the RNA, from the metabolites, and all pooling at one place, multi-domain, multi-genome knowledge base. Then integrating this data and making an meaningful result so that we can avoid further animal testing. So very quickly we'll go through here. See, data from experiments that evaluate the effect of a chemical on global patterns of gene expression, that is transcriptomics. Okay, protein content, that is proteomics, and small molecule and metabolites. See, the question is related to here, that is metabolomics as well as metabolomics combined with genome information from both the test species, that is rat and mice, and target species of interest, example humans, are analyzed by computational tools, that is bioinformatics. So many things you are integrating together to get a meaningful information per unique or potentially predictive patterns. So algorithms will be written here of the toxicity essential to the use of omic data For predictive toxicology and safety assessment is the ability to reliably the observed omic patterns for traditional measures of toxicity such as histopathology, clinical chemistry, etc. So before doing these animal trials, this kind of integration of data can be very much useful at the initial stages of the drug discovery. So if you can see, this is you're doing histopathology, clinical chemistry, your weight of evidence, physiology you're looking into, then you're looking into PK, that is ADME, okay, your gene, protein, and metabolite expression profiles, your omic database, genome database, both are going to computational analysis. And I told you, know, here multi-domain and multi-generation knowledge base, okay. You're also looking into literature mining. If any read across data on that investigational chemical, then everything you're putting it in a single page, box. Okay, identifying gene, protein, functional group, pathways, and networks, and toxicology database, that information also. Then finally, what you're doing, you're going to integrate and make a meaningful information out of it. This is the application of this omics in the case of toxicology. So please go back and check this page number 49 and Clason's 9th edition. So it regarding omics, it is mentioned in ABT brochure two places under design executive toxicology also, as well as in the case of interpretation also. So check those things, part of syllabus it is. Now we'll go into second question. Second question is, all of the following are true concerning the animal cancer carcinogenicity studies except. So when it comes to animal carcinogenicity, generally it is 2-year mice or rat, okay, or 18-month mice say. And there is another to cut short the duration, we do transgenic mice assay, 6 months transgenic mice assay. Okay, so with that, one question is there. Let's see. Historical control data is never considered. So this is the wrong option. Generally, in the case of this 2-year cancer study or any chronic toxicology study, no, always will check for historical data. So some of the findings, some of the adverse effects, are they seen in the case of historical control group in that particular CRO? Like that, we'll be checking. Then we can avoid that particular adverse effect, not consider it as a serious adverse effect, based on which you need not derive NOEL or NOEL. Okay, so rest of the options like B, C, and D are right with respect to which are applicable to animal cancer study. So second option was a rodent cancer study usually takes 2 years. As I told you, generally rat is 2 years. Certain times mice 2 years or 18 months you'll come across when you look the literature for these for the chemicals in toxicology literature. It is impractical to use doses that would occur in normal human exposure due to large number of animals required. That is completely true. Around, I think, 50 animals per sex per dose or something like that, or 40 animals per sex per dose. So go back and check OECD guide. Yes, we have here, OECD guideline 451 is the one which is responsible for this 2-year cancer study. And here you can see the species is either rat or mice, and number of animals you see 50 to 65 sex per dose. Okay, so huge number of animals are needed in order to carry out the 2-year mice or rat rodent assay. Gender differences in background tumor incidence are sometimes observed. Yes, there are certain times the Noel for female rats is different, Noel for male rats is different. There are certain findings, tumor findings, which are only seen in the case of male rats, or it is not seen in the case of female rats. So gender differences also plays a very important role, because you're dosing the animals for a long period of time also. And so that is what, so I would request you to pause here and check what are the different OECD test guidelines which are required for cancer study. We have, as we told now, we have OECD 451 guideline, and we also have OECD guideline, test guideline 453, in which combined chronic toxicology as well as cancer study. So within this repeat dose toxicology study, we're also integrating cancer study. I think there will be interim sacrifice. Some of the animals will be continued for 2-year dosing, and some will be interimly sacrificed maybe at 9 months or 1 year, right? So pause here and see what are the observations in these studies and what is the duration of the study. Under this table, they have also given chronic toxicology study. Even though it is not a cancer study, still why they have given here it is, try to understand. Many a times from these chronic studies, either it is 6 months or 9 months or 1 year chronic study in the case of rat or mice like that, you can we observe for pre-neoplastic lesions. If there are any pre-neoplastic lesions, that also gives an indication that that particular experimental chemical is having a potential concern for causing cancer. Okay, pause here and try to remember. Now we'll go through question number 8. Question number 8 is examples of significant concentrations of a toxicant in a tissue that is not a target organ include all of the following. So the right answer will be in the green text, that is paraquat. So the question is we need to identify the toxicant, the chemical which is responsible for causing toxic effects, and the tissue or organ or target organ we need to identify. So the paraquat, no, it will go and it will go and deposit in the lung. It is going to concentrate in the lung. Paraquat, and along with that paraquat also read diquat. These are the two herbicides, and the chemical group is bipyridal groups. Okay, so what is happening in the case of paraquat is the intention is not to go and deposit in lung, but unintentionally the person who is applying it will to that applicator, to the farmer, this will go and concentrate in the lung, and it will cause severe lung necrosis. So free radical formation will be there, redox reactions will be there. So pause here and go through. So some questions in the ABT, they have frequently been asking about paraquat. The target organ will be here, lung toxicity. Whereas in the case of another chemical in the same class, that is diquat, diquat is also very potent, very highly toxic, but it is going to affect more in the case of GI tract. So this question they will be asking you. They'll try to confuse you in the exam with the options. Okay, and next we'll quickly go through other options which are right options. Lead. So whenever a person is exposed to a lead, so major concentration more than 90-95% of the lead, this is a metal element or whatever it is, it will go and deposit in your lung. Coming to DDT, which is highly lipophilic. So with respect to DDT, also read about silent spring. So it is going and depositing in the adipose tissue. So because of its lipophilic nature, this DDT will go and accumulate in the deposit in the fat tissue. So what happens, whenever you're consuming for example a food which is contaminated with DDT, or animals are there, grazing animals or like that, so they or birds, they consume grains which are highly rich in DDT, so this will go and deposit it in their bone. So during winter, during hibernation, uh, the slowly fat start dissolving because you are not consuming food. So DDT concentrations in the plasma or in the blood will increase, and thereby it will cause toxicity. Even though at that particular point of time he is not consuming DDT, but DDT is stored in your body in the fat. Fat will slowly get dissolved when the energy crisis is there, particularly during winter hibernation. So at that time, excess concentrations of DDT in the plasma or serum will cause toxicity. Lipophilic nature is the reason for it. And another thing is option number D, that is TCDD, Agent Orange. So with respect to Agent Orange, we have already uploaded one video in our channel. Coming to TCDD, this is also because of its lipophilic nature, it will go and bind to the adipose tissue. So very very important. Which drugs will go and bind to bone or a heart? So their location site is very very important. So one question with respect to tissue distribution or tissue deposition, like that they will be asking in the exam. Okay, so paraquat, diquat, read read about DDT, that is silent spring, and read about Agent Orange, lead also, and also see what are the other metals which go and deposit in the bone. Okay, minerals or metals. Because okay, now we'll enter into question number 9. Question number 9 is ability of a chemical to cause acute skin and eye irritation is usually evaluated. So what you need to identify the test species. So what are the test guidelines which are responsible for checking this acute eye irritation and acute skin irritation, and also we need to check what are the test guidelines. So as per OECD guideline, that answer will be the green test, that is rabbit. So rabbit is the test species in both, which is used for both identifying skin irritation, acute skin irritation, as well as acute eye irritation, and the test guidelines are OECD guideline 404 and OECD guideline 405. So frequently they'll be asking acute toxicology studies. So six pack study will be there. Here you can see 404 as well as 405 is there. So other things 40 skin sensitization will be there. Okay, so these guidelines which are responsible for causing this acute toxicity, please remember and read them. Okay, in detail. Some other video will prepare in detail about 404 or 405. Okay, so next question is, before a potential pharmaceutical compound can be given to human, So what type of filing we have to do? So with respect to filing, we have discussed some in our channel, some of the videos. I will give the description in the link in the description. So the answer will be before starting phase one, or we need to file IND. So people, try to remember the drug discovery pathway. First is IND, then NDA. So once the patent expires, then you will go for ANDA. Okay, and this is for conventional molecules. Again, biological molecules or biopharmaceuticals is completely different. And another thing that you have to remember is what type of filing. Is it 505B1 filing, 505J filing, 505K filing? That type of filing also you have to remember. So right answer will be IND. Don't get confused. When you see in the exam, you'll think that option A is right. NDA is correct, new drug application. But still, the first initial no pharmacology, efficacy, safety pharmacology studies, or initial pilot studies, initial short-term studies, all these things you will give in the form of IND. Then lot of pre-IND meetings, type A, type B type of meetings will be taking place in between the FDA as well as the sponsor. Based on that, they will decide next application will be new drug application. So don't get confused. Coming to option C and D, acute toxic studies on 4 species must be conducted. There is no need of compulsion. Must even ethics committee will also not request you to do it in 4 species. And 2-year dog cancer study must be completed, as we have seen in the previous slide. Generally, for cancer study, the rodents, that is mice and rat, are the test species. Okay, you have to also understand we cannot have huge number of dogs to conduct these studies, and that too for a 2-year study. Okay, next question is phase 1 clinical trial are conducted to determine all of the following. So what is the main intention of phase 1 clinical trial is to identify pharmacokinetics, to identify safety and efficacy. So rare type of adverse effects, generally we will do it in phase 4, that is pharmacovigilance or post marketing trials. Okay, so some questions with respect to regulatory submissions or drug discovery they will ask. So initially you have seen some questions with respect to proteomics, metabolomics they were asking. Then type of filing you are seeing. Now they are asking what is the important significance of phase 1 clinical trial, like that. Okay, next question is MTD stands for. MTD is nothing but your maximum tolerated dose. So try to go back and check MTD in which type of studies you are doing. See, you will come across MTD in your acute single dose toxicology study also. Objective will be MTD. In the case of repeat dose also, you will be deriving MTD. So generally, objective will be NOEL, but you can come across certain reports, study reports, where MTD will be derived, and also in the case of cancer studies also will be deriving. In addition to NOEL for tumors and non-tumor effects, you will also come across MTD. So try to read the MTD definition with respect to all these three type of studies. Okay, the acute toxicity studies in animal provides. So main objective of conducting a single dose toxicology study or acute toxicology study is to identify approximate lethal dose, that is LD50. So for that we have OECD guideline 420, OECD guideline 423, as well as OECD guideline 425. There was initially OECD guideline 401, which is absolute, which is deleted from the test guidelines, sir. Next, information on the target organ. Generally, identification of target organ is generally it is the objective of 28-day repeat dose toxicology study, but also you can identify, you can have an idea from this acute single dose toxicology studies. Okay, so if you know the death of the particular or the morbidity of a certain animal is because of the respiratory system or because of the liver or kidney, like that you can identify the target organ. Now next is information on dose selection for longer term studies. Yes, this is also there. Once you identify MTD or you identify LD50, fractionation of those doses can be used to decide the doses for next 14-day or 28-day, like that. Okay, so all of the above is the right answer with respect to question number 13. Now we'll enter into question number 14. Question number 14 is a subacute toxicity studies in rat usually last. So some questions with respect to study duration they will try to ask you. So here the right answer is 14 days, but I would request you go back Clayson 9th edition, page number 33. In this diagram very clearly they are given. So whenever there is a single exposure or multiple exposure within 24-hour timeline, that will be considered as that will be considered acute. Okay, subacute is less than 1 month. Subchronic is 1 to 3 months, and chronic is more than 3 months, that is 90 days or up to 1 year. Okay, so don't get confused with the English this thing. You'll be thinking acute, then subacute, Then chronic, subchronic, you'll be thinking it is not like that. They will try. This is always a confusion. When it comes to English, you'll think that acute sub, then chronic, then subchronic it should come, but that is not true. Try to remember this thing. So one question with respect to study duration they'll be asking in the exam. Next is the period of organogenesis in rats. So with respect to whether it is fertility, early embryonic developmental study segment 1 or segment 2, that is embryo fetal developmental study. They'll try to ask you question with respect to organogenesis period or gestation period. Here the right answer is 7 to 17. In next slide, we'll discuss in detail about rat organogenesis period as well as rabbit organogenesis period. Next, we'll enter into next question number 16. A dose of investigational drug that suppressed body weight gain slightly in 90-day animal study is defined by some regulatory agency as MTD. As I told you, no, MTD you'll get it from single dose toxicology study, also from the cancer. Few cancer reports are there, study reports are there, where you'll come across MTD in addition to NOEL for tumor effects or non-tumor effects. So similarly, in the case of 90-day study also, based on this decrease in body weight, they have defined MTD. So the right answer is the green text, MTD. Certain regulatory agencies for an investigational drug suppress in body weight gain slightly in 90-day study is also defined as maximum tolerated dose. Okay, so very quickly we'll go through what is MTD definition. Maximum tolerated dose is the highest amount of a drug or a treatment that can be administered without causing unacceptable severe side effects or toxicity. So you're getting there shouldn't be without causing unacceptable severe side effects or toxicity. It should not cause any severe adverse effects. So that particular dose will be considered as maximum tolerated dose. It is a clinical benchmark in the case of pharmacology used to balance between maximum therapeutic benefit against patient tolerability. So that is the word it is coming from, maximum tolerated dose. Pause here. As I told you, try to compare with these three studies. What is the MTT significance in these three type of toxicology studies? Now we will enter into question number 17. 17 is a subchronic animal studies required by the FDA will usually include. So subchronic, as you have seen, it is 1 to 3 months. Okay. So two species, usually one rodent and one non-rodent. That is two for conventional molecules. You will do either one rodent, rat, mice, or you will do it here in the case of non-rodent, rabbit, dog, or monkey. Both genders, that is males and females, at least three doses group: low dose, intermediate dose, mid dose, and high dose. So this is a typical 90-day study. You will be having males and females, both the genders you will be testing. Low control, low dose, mid dose, and high dose group will be there. Okay, so next, we will be entering into question number. In continuation, as we discussed now, we'll discuss in little bit with respect to gestation, because this is a critical window period where certain drugs, certain chemicals, experimental chemicals can show teratogenic effects. So to understand what particular window period that we need to see, that we have to see. So question was with respect to rat organogenesis and rabbit organogenesis. The gestation period is, in the case of rat, is 21 to 22 days, whereas in the case of rabbit, the gestation period is 30 to 31 days. And organogenesis, this is a critical window at which you will be finding teratogen. If a chemical is exposed during this particular window, you can correlate it with thalidomide. During this particular critical window only it was exposed. So in the case of rat, it is GD 6 to 15. So most of the EFD studies in rat, if you see, the treatment duration will be gestation day 6 to 15. Whereas in the case of rabbit, some studies you will see GD 7 to 18, and some places gestation day 7 to 19 you will be coming across. When you do literature search now, you will come across these durations. Next, what are the important outcomes, key milestones in this is, during this particular rat organogenesis series, this is when the ectoderm, mesoderm, and endoderm, these are the different layers. From the blastula, gastrula stage will be there, so these are the three layers: ectoderm, mesoderm, and ectoderm differentiate into vital internal organs. What are those organs? We'll see. Heart, the brain, lung, like that. So we'll see. Early somites form, neural tube closure occurs. Okay, there will be a closure of this neural tube, and the heart, brain, liver, and limb bunds rapidly develop. So these, during this period, heart, brain, liver, and limbs will be developed. That is the reason thalidomide was, when it was exposed during this particular window period, phocomelia were there, short limbs were there. Okay. Well, coming to rabbit, implantation typically occurs during GD 6 to 7, immediately followed by gastrulation and organogenesis. To understand this gastrulation, you have to see these stages: blastocyst, gastrocyte, and organoid. Okay, followed by gastrulation and organogenesis. During this period, this particular organogenesis period, in the case of rabbit, the major organ system include heart, that is cardiovascular, respiratory, lung, and CNS differentiation, and they start taking the shape. So pause here, try to compare, see these guidelines: OECD guideline 414, OECD guideline 421 and 422, combined reproductive developmental toxicology study 443, as well as ICH S5 guideline, which is for reproductive toxicity. So in the ICH S5 guideline also, around 15 drugs, I think they have given listed out, which will cause teratogenicity, and you have a separate point of departure in that. In that, they will give you MEFL, maternal effective fetal developmental level. NOELs will be listed. So those drugs which are listed on ICH-S5 guideline, no, they're very, very important. So go back and check those tables. Yeah, I will also try to give the link in the description. I have listed out the ICH-S5 tables also, so you can quickly go through. So next, coming to last slide, we are discussing question number 18. Question number 18 is something related to kinetics. Okay, so and receptor binding. When all the receptors are occupied by a toxicant. So now they are talking about saturation of the receptors, binding of the receptors, and there is a maximum amount of receptor toxicant complexes. The response is labeled Emax. So you have to here quickly you have to refresh your memory with respect to what is Cmax, what is Emax, what is Tmax, what is half-life. So some questions, if with respect to kinetics, if they ask in exam, you will be able to address. So in pharmacology, Emax represent maximum achievable effect or maximal drug response a system can produce. For example, the system can be either human or a rat, or in the case of mice, like that, when its target receptors are fully bound. So whatever the concentration of the drug you are giving to this system, for example, mice or rat, complete binding of the drug, so to the whatever the receptors available in that system should be there. Okay, fully bind. When its target receptors are fully bound, the theoretical ceiling of a drug activity, increasing the drug dosage, even once all the receptors are bound by this particular drug, even though if you increase the concentration of the drug, there are no free receptors available in that particular test system, that is mice or rat. So there will be no meaning of increasing the so these all things you'll come across in the kinetics, linear kinetics, what is a straight line kinetics, first, second, third kinetics will be coming across, no. So increasing the drug dosage beyond this point will not increase the physiological or biochemical response, because all relevant receptors are saturated. So saturation of the receptors is again very key concept. Next, we'll see question number 19. So, which of the following statements are true? So, all these statements are not true. So, what is that? The toxicant receptor interactions are always reversible? No. There are certain, see, binding of these receptors, also different types of bonds will be there. Ionic bond, covalent bond. So, always it is reversible. That is not true. Certain times it might be irreversibles, or Van der Waals type of binding will be there. So, you have to think these things and try to read. Second question is the second option is receptors for toxicants are always enzymes. No, the nature of the receptors might be enzymes, proteins, or certain times some functional groups will be there. So that is also not true. The toxic responses is related to toxic concentration in the plasma more so than the concentration at the site of action. Once we never know that one, but the moment you consume a certain pill, a certain drug, so how much amount is there in the plasma, how much amount it is going and binding to the particular target site, this is not always constant, and it is not more than so than concentration at the site of action. That this statement is not true. So now we'll see question number 20. So question number 20 is an increase in free drug concentration will increase the pharmacological effect. So as I told you, this particular slide is only with respect to kinetics only. So free drug concentration, as we have seen in the previous slide, drug, if it is going and binding to the adipose tissue, so there is no in the case of that DDT, as well as in the case of TCDD, if it's going and binding to the fat or adipose tissue, there is no free drug available in the plasma to go and bind to the receptor or bind to the target receptor and show the efficacy. Okay, so Increase in the free drug concentration will increase the pharmacological effect, decrease the toxic effect, decrease the amount of drug filtered at the glomerulus. So these options are not applicable to the first statement. Pause here, try to read. So from this series of questions, like 6 to 20 questions, we discussed lots of concepts. We try to revise them so that frequently these related concepts they'll be asking in the exam. See, we have discussed about metabolomics, proteomics, tissue distributions. We have discussed acute toxicology, MTD, study duration. So these are the basic things that you will come across, particularly in the first four chapters in the Clayson textbook. That is principles of toxicology. Either it is risk assessment. Also, we have seen. We have seen ADME, that is tissue kinetics also. So from this, they will be asking you. Okay. So thank you so much. Be curious. For any questions, you can always write a mail to arya.priyanka@gmail.com. All the best for your exam.